Hi, my name is Sarah, and today we'll be talking about post-translational glycosylation. By the end of this lecture, you'll be able to explain what post-translational glycosylation is and identify its purpose. You'll be able to recall the possible biochemical pathways and describe the proteins and macromolecules involved. And finally, be able to explain the overall significance within the cell. Post-translational glycosylation is the formation of conjugates between polysaccharides and proteins. So what it does is that it adds saccharides to specific protein residues in order to create an optimal structural stability as well to improve the function of the protein. There are three different categories of conjugates. The first one is the proteoglycan, which are unbranched polysaccharide chains formed by repeated disaccharide units. Another one is the peptidoglycan, which are polysaccharides linked to small peptides that form large cross-linked complexes. Now the last one is probably the most common one, and that is the glycoprotein, which are proteins that contain oligosaccharide chains covalently attached to amino acids. One thing to remember is that this process occurs in the rough endoplasmic reticulum, in which it is further modified in the Golgi complex. One thing to note is that post-translational glycosylation plays a major role in cell-to-cell recognition process and in signal pathways.
Okay, so there's many different types of glycosylation, and there's 5 common ones that we're going to discuss in this presentation. The first one is the most common, and this is N-glycosylation. It's when a carbohydrate is added to the nitrogen of asparagine or arginine side chains, and this affects the glycoprotein folding in eukaryotes and allows cell-to-cell and cell-to-extracellular matrix attachments. Next is O-glycosylation, which is when a glycan is added to the hydroxyl of serine, thronine, tyrosine, hydroxylsine, or hydroxyproline side chains. This occurs in eukaryotes in the Golgi apparatus, in archaea, and in bacteria. The third type is glycation. This is a carboxylic acid replaced by a GPI anchor, and then it links proteins to lipids through a glycan linkage. The fourth type is C-glycosylation, which is when a sugar is added to the tryptophan side chain, but this is pretty rare, and usually it's a mannose sugar that's added. Lastly, we have phosphoglycosylation, which is when a phosphoglycan is linked through a phosphate on a phosphoserine. So on this slide, you can see the biochemical pathways for each of the processes I just told you about. Each one has a unique protein that allows it to happen. So N-glycosylation, being the most common, is probably the most important for you to understand and recognize. And so the protein that allows this to happen is OST-ACE. And then for O-glycosylation, you're using the protein GTFS. In glycation, you're using GPIT. In C-glycosylation, you're using CMTFS, and then in phosphoglycosylation, you're using PTACE. And so in this image, you can sort of see exactly what groups are being replaced. So for example, one of the hydrogens is replaced with the carbohydrate in N-glycosylation.
Hello, my name is Dirk Bates, and I'm going to be talking about the significance of glycosylation for the cell. Glycosylation affects protein folding, conformation, and stability of the cell. It also increases the proteome diversity, the most out of any form of post-translation modification. Glycosylation will also affect about 50% of all of your proteins in the human body. Glycosylation, when it comes to protein folding, plays a huge role because there's a good majority of proteins that will not function without glycosylation occurring because the fold to make that function possible will not occur. The efficiency is also a key factor of glycosylation because we're going to decrease the amount of resources used and increase the time in response to stimuli. This is done by adding a quick modular switch as a subunit that can turn on or off a function rather than having to build and break down a new protein through synthesis every single time. We are also going to have some cell-to-cell interactions that occur because of this. An example of this would be adhesion via sugar-binding proteins called lectin. This is actually a response elicited by the immune system. And speaking of the immune system, viruses also use glycosylation as a shield on their coating to protect themselves from our own immune system. And our last little tidbit for the significance, in humans at least, is the presence or lack of presence of glycosyltransferase. This dictates which blood group antigen you will receive with your blood type when you are born. Well, thank you all for listening. Hopefully you have learned a bit more about glycosylation. Stay safe, y'all, and have a fun time in quarantine.